Now, one of the things people kind of have wondered is why the hell are you looking in the Berkeley pit for compounds with biological activity? We've looked at a lot of Native American medicinal plants. We've looked at Ayurvedic plants. We've looked at Chinese medicinals. And those are very rational places to look for drugs because they have, in many cases, 5 to 10,000 year histories of helping people deal with diseases. Why would you look in a toxic waste dump for a fungus to help you with disease? Well, it turns out if you're a little fungus living in the Berkeley pit, you have found a way of dealing with low pH, so a very acidic condition, and a high reactive oxidizing environment. And it just so happens that low pH and reactive oxidizing conditions are the hallmarks of a number of different pathologies within cellular systems. As a matter of fact, cancer can be very reactive oxidizing, and it can also have a low pH. Inflammation is highly acidic. So those actually gave us a little bit of a comparator to look at. As we started looking at specific enzymes, turns out a number of the enzymes that are produced by fungi are very analogous to enzymes in human systems. Two of those are caspases and matrix metalloproteinases. And so some of the same compounds that these enzymes work on are common to both human systems and fungal systems. Some of the same compounds that inhibit them can be common to both. So there's a connection there between the conditions in the Berkeley pit and the conditions in a pathologically deranged human cell. And so we sort of went from a bio-irrational to a bio-rational approach because these same fungi, whether they're found in a human or found, these same enzymes, whether they're found in a human or a fungus, can actually respond to the same types of chemistry. So our eureka moment number one, we stopped looking for antibiotics because in our first studies, we found absolutely no antibiotic activity with any of our fungal preps. And if you think about it, even though we're finding life in the Berkeley pit, that life is pretty sparsely spread. We don't have a rich slurry of fungi and bacteria in the pit. You know, you take a soil sample and it's teeming with microbes. The Berkeley pit, if you take a cup of Berkeley pit water, there might be 1,000,000 fungal spores in there, which is pretty sparse indeed. So those organisms don't have to fight each other. They do have to fight their environment. And so we started looking at enzymes that were compatible with the idea of fighting low pH and reactive oxidizing conditions. So I'm going to start with our favorite enzyme, caspase 3. Caspase 3 is the enzyme that's associated with programmed cell death. It promotes the death of cells. Now, in a deleterious sense, caspase 3 is produced at very high levels following a stroke. So if you have an infarction, you have about 4 hours to get to a hospital and take the only drug that is currently available, and that's a clot buster drug called TPP. If you have a different type of stroke that doesn't involve a stroke, a clot, but is a bleeder, there's nothing for you. Now, following that infarction, if you look at the infarction zone of the stroke, for the next 48 hours, those brain cells are pumping out caspase 3. And the goal is to kill a huge region around that infarct in order to, in essence, protect the rest of the brain. But in doing that, you are losing large amounts of brain tissue that do not need to be sacrificed to that infarct zone. So our goal, what could we find that might inhibit the production of caspase 3 in newly infarcted brain tissue? We worked with a wonderful collaborator here at the University of Montana, Daryl Jackson, and we were looking at studies on how caspase 3 mediates neuronal death following an infarction. It also turns out that caspase-3 is involved in the onset of Alzheimer's, but that's a different application. And so looking at this incredible interaction of caspase-3 and healthy brain tissue, I'm going to show you one of these pathways I talked about with enzymes. They're very complex. So this is a brain cell. You have some sort of external stimuli, and it's causing a whole cascade of reactions. You don't even need to bother reading the titles until you get to caspase-3. Caspase-3 is that sentinel enzyme that actually goes into the nucleus, causing DNA damage and cell death. And this is what we wanted to stop right here. This pathway is extremely important, and is there a way of stopping it? So we actually had an enzyme-based assay where we could take caspase-3, take our test compounds, actually go all the way from crudest extract all the way to pure compound, and see if there was some inhibition of the action of caspase-3. And you can buy these beautiful little test kits that allow you to do this with a fluorescent reporter at the end. So these types of assays are very powerful tools for isolation. These are just some of the lovely compounds with amazing chemical structures produced by different fungi that we discovered in the Berkeley pit. A lot of different shapes. And if you look at a molecule like this, you're looking at each point, each one of those represents a carbon atom if you're not familiar with the chemistry. My apologies to Dick Field, who is a phenomenal chemist, but I don't know if everyone is comfortable with structures like this. So most of the molecules we look at as organic chemists, they're composed of carbon, hydrogen, and oxygen, nitrogen sometimes thrown in, and the occasional sulfur atom. And these beautiful molecules then will fit into the active site of an enzyme like a caspase-3 and inhibit its activity. A second enzyme that's extremely interesting to us is a cousin of caspase-3, and this is caspase-1. Runaway inflammation is the hallmark of caspase-1. Now, it's an important enzyme. You need some inflammation, but when it goes amok, you start seeing pathologies like multiple sclerosis, Alzheimer's, myelogenic leukemia, Huntington's disease, type 2 diabetes, and a whole array of other pathologies. Again, a complex pathway associated with caspase-1, and it's actually kind of a beautiful dance of molecules within the cell. But when an individual macrophage, now this represents a macrophage, part of the immune system in a cell, when it is stimulated by, remember these PAMPs and DAMPs, danger and pathogen associated molecular patterns, they start a cascade that ultimately results in the activation of caspase-1, the enzyme we're interested in, that will then take precursor cytokines like pro-interleukin-1 beta, pro-IL-18. These are very important in enhancing inflammatory pathways, and start activating them. And if they don't stop, if they don't turn off, then we just start pumping out pro-inflammatory cytokines. So we wanted to find a way of taking caspase-1 and turning it off. And so we used the same enzyme-based type of approach, looking for compounds that were capable of inhibiting caspase-1. And we found about 53 of them, also some very interesting structures. Our final enzyme that we care about is an enzyme called matrix metalloproteinase 3, MMP3 for short. Matrix metalloproteinase is a very important enzyme that is tied into wound healing. So if you do not have MMP3 and somebody slices you open with a knife, that wound simply will not heal. Your tissue, the epithelia on both sides of that knife wound, have got to dissolve into a slurry, migrate back and forth across the wound, and then reform as a new epithelium. That's kind of the basis of healing. But if we have too much of that slurry formation, then pathologies can ensue. And it's very important in metastatic cancer. So, to give you a cartoony version, there's an important protein called E-cadherin. E-cadherin is the cement that holds our cells together side by side. MMP3 cleaves that enzyme. So the cells start moving away from each other, but they're still held by this sort of basal lamina. So MMP3 does something else. MMP3 turns on another enzyme called matrix metalloproteinase 9. MMP9 dissolves the basal lamina. So now we have this MMP3 dissolving cells, activating another enzyme that helps dissolve cells further. It actually acts in a pathway approach where it blocks the production of more E-cadherin. And so MMP3 is simply working to turn your epithelium into a slurry. If this is happening in a cancer cell, what it's doing is it's mobilizing a solid tumor and turning it into a highly migratory mess that can now migrate through your lymph system, through your blood system, and then reach another target. At that point, that slurry has to invade the distant target, whether it's lung, bone, brain, a lymph node. And invasion is another aspect of this type of process. So when we have something like MMP3, it's allowing the migration and the invasion of cancer cells. We want to stop that. And so again, we found these complex molecular pathways. And something else we found, it's highly connected to inflammation because it turns out Runaway's caspase 1, remember our pro-inflammatory enzyme, also enhances the production of matrix metalloproteinase 3. And so if you have runaway inflammation, I was going to say runaway inflation, I'm sorry, that's a different topic, then you're going to start having runaway production of this pro-metastatic inducer MMP3. So if we can block caspase 1, we can also help mitigate metastatic spread in cancer. And so this was sort of our eureka moment number 2. There are papers galore that talk about each of these pathways, but the connections are few and far between. But we recognize that through these reactive oxygen species, which stimulate both pathways, that inflammation and metastasis are highly connected processes. So why do we care? Why would it matter to us if a solid tumor dissolves, turns into a slurry, and moves to a distant site? We care because the National Cancer Institute demonstrates quite dramatically why we care. These are the 5-year survival rates for different types of cancers. If the cancer is local, regional, in other words, it's only moved to, say, a sentinel lymph node, or metastatic, moved to a distant site. If you are a woman with breast cancer and you have a local tumor, you have a 99% 5-year survival rate if properly treated. Once it moves even to a regional lymph node, your chance of survival drops about 15%. Once it metastasizes, it's a beast. And that beast is very hard to kill. For my husband, Don, and I, these next 2 cancers are near and dear. I had ovarian cancer back in 1999. I'm happy to say I'm a 21-year survivor. Mine was highly localized. I had surgery and nothing else. And 20 years later, I'm not worried about its recurrence because it never made the move to being a metastatic ovarian cancer. So when we were working on Taxol, it was particularly poignant because this was about the time that we were working on Taxol was when I was diagnosed. Don more recently was diagnosed with a colorectal cancer. Again, he was fortunate. It's very localized. His doctor tells him he has a 90% chance of non-recurrence. although as you can imagine, we're very vigilant about it, as opposed to a 13% chance of survival once it's metastatic. And go down and read these numbers. Horrendous chances of 5 year survival once you hit a metastatic spread. Now, every day we're coming up with new anti cancer agents, and they're doing amazing things, including the new biologicals. But if you can target a cancer before it spreads, your survival is so much more assured. Again, we found a number of compounds, and with the help of colleagues at Memorial Sloan Kettering, Eisai Pharmaceuticals, and National Cancer Institute, part of the National Institutes of Health, we were able to test all of these compounds that we had discovered because of their ability to inhibit MMP3, tested in a number of different human cell line trials. So this is just a partial list of some of the compounds we isolated. This is a list of some of the cancer cell lines against which we were tested. And these are some of the what are called IC50s. So these are the doses that could use to inhibit the growth by 50%. So these are looking at growth inhibition as opposed to actually cell death. And some of these compounds were actually active at the nanomolar level, which is clinically relevant, and some of them at the low micromolar level, which is approaching clinical significance. Um, and with just a little bit of tweaking, some of these compounds actually have the opportunity to move towards clinical trials. And so it's a very exciting validation of the type of approach that we've been using, where compounds that show activity in enzyme assays can then translate that relevance into cell line activity. Now, I mentioned invasion and migration. We can actually directly evaluate the ability of a compound to prevent invasion, migration of these cancer cells through a matrix, and then the ability to actually invade a second matrix. And we had the privilege of having a wonderful, actually a high school teacher with an amazing background in science, Lily Appadale, worked with us for 2 summers, funded by the MJ Murdoch, getting my foundations mixed up, MJ Murdoch Foundation. And she was looking at the ability of these compounds to block migration and invasion of highly metastatic breast cancer cell lines, pancreatic adenocarcinomas, and lung cancer adenocarcinomas. And her work was really remarkable and very exciting. Um, just to give you an example of how structure and activity can vary activity dramatically, this is one of the families I showed, the burkitoazophylones. These compounds will often have the precursor Burk in their common name because they came from the Berkeley pit. So burkitoazophyllone A, B, and C, very similar compounds, but the activities are profoundly different. Burkitoazophyllone A has a very low activity, low ability to inhibit MMP3, and no activity in a human cancer cell line. The central compound B, which only differs by this little 3-membered oxygen-bearing ring called an oxirane ring, the only difference between these two molecules. And the compound is an order of magnitude more active than its cousin, and is extremely active against a number of cancer cell lines, particularly leukemia and melanoma cell lines. And if you get rid of this ring entirely, you get rid of all activity. So those types of structure activity relationships can now help us as chemists figure out what can we do to this compound to enhance its activity. And you can be assured we're not going to open that ring, because if we open this ring chemically, we're going to get rid of all activity. So that's also an important part of what we do. So eureka moment number 1, chemistry of the extreme environment directs the secondary metabolism. And of course, we expect no antibiotics because of the sparse density. And so microbes do not produce antibiotics if they don't have a reason to. If there's no competition, why waste your energy killing other microbes? Eureka moment number two, inflammation and epithelial mesenchymal transition are intimately connected. Eureka moment number three, always expect the unexpected. There's a thing called cryptic biosynthesis. A lot of organisms produce compounds we'll never know about because we don't give them the right growth conditions. So cryptic, it's hidden. And what can we do to turn it on? Well, it turns out we grew two of our fungi together and we had our third eureka moment. These fungi produce some amazing antibiotics if you give them the right stimuli. So I'm going to introduce our last molecule in the series. This is a new molecule and actually a new family of antibiotics called the Berkeley lactones. Lactone is a ring molecule that's held together by this little ester function. And they're fairly simple molecules that are distantly related to bacterial lactones, like erythromycin, although their pathway is actually a little different in how they're formed. These molecules, when we just thought we'd test them because they superficially resembled an erythromycin, we tested them with the help of Nigel Priestley's lab here at the University of Montana. And we found that particularly this molecule we call our lead compound is extremely active against Staphylococcus aureus, kind of a wild type, plain vanilla variety of Staph aureus. Closely related analogs, so this is a family of compounds, also showed activity against Staphylococcus aureus, and some of them showed absolutely no activity at all. So again, there's a connection between specific structure and activity. And what we found was that we need to have a ketone moiety here, an ester moiety here, and this strange little side chain in order to have the kind of activity that we're elaborating. Now, Nigel Priestley has had a long history working on antibiotic isolation and characterization, and he has colleagues at the University of Connecticut at an infectious disease specialty, and his collaborators have given him about 40 different drug-resistant Staph aureus. So these are MDRSAs, they're multi-drug resistant Staph aureus. And these are the beasts we're trying to kill. And so Jeremy Alverson, his lab technician, started testing our compounds, and now we have quite a nice collaboration. And it turns out our Berkeley lactone A is actually more active against drug-resistant staph than it is against wild type. Now, I'm not showing all the data here, but it turns out we're also active against Bacillus anthracis, which is an important bioweapon bacterium, and Mycobacterium tuberculosis, which is a very hard bug to kill, and of course causes tuberculosis. Nigel has actually tested all of these different well-known antibiotics against these same isolates of Staph aureus. And this is looking at the 99% kill rate of our compounds, so Berkeley lactone A. We're killing these compounds at 1 microgram per mil. Our closely related second most active compound is about 6 micrograms per mil. And if we look at linezolid, vancomycin, erythromycin, clindamycin, levofloxacin, we're looking at their kill rates. Vancomycin is a little bit better than we are. Vancomycin is the result of years of study and optimization. This is what Berkeley lactone A looks like right out of the fungus without any optimization at all. So we are extremely excited about this new class of antibiotics. And One of the things we now have are grants from the National Institutes of Health to study these organisms, to study these antibiotics. And recently, the DoD just granted us a nice little grant to study mechanism of action and application. So we contend, even though our geochemist colleagues might disagree with us, that our microbes could be the richest ore ever mined from Butte's richest hill on Earth. And so I'd like to thank, we've had a number of funding sources, but not always. We've had some really lean years. And during that time, Don and I created our own foundation here at the University of Montana. And we did it so that we could donate to ourselves because it was the only way we could keep the lab open. And in that time, somebody else donated $40,000 to us. So that anonymous donor, we later found out who they are, and they've become very dear friends indeed, very generous and loving people. Some great colleagues here at the University of Montana. Terry Gertzman, who was a postdoctoral fellow here, she worked with us on the inflammasome in caspase 1. Daryl Jackson, who's now in Spokane, worked with us on caspase 3 and brain infarctions. Jay Evans, who is at the new Inimmune Corporation here at the University of Montana, formerly down at Rocky Mountain Labs. He and Nigel Priestley in the Department of Chemistry are working with us on the antibiotic project. Bureau of Mines gave us all of our water samples in the Berkeley pit, and we thank them because without them, we would not have had a source. And we thank, like I said, we've probably had 100 different undergraduates who have moved through our lab. Now, many of them are doctors, veterinarians, dentists, school teachers, wonderful citizens, and many of them are very dear friends. So we have had an amazing run of it. We continue to work with these lovely little creatures. And hopefully we're going to find one of these days that one of these compounds will actually cure something and not just be something that we love.